This flagellum, which is present outside the cell, may be absent in certain bacteria. That means few bacteria have flagella and few bacteria do not possess this flagella. A single one is called as flagellum and plural of that is flagella. The bacteria can be divided in two categories depending on presence or absence of this flagella. The bacteria which possess this flagella, they are able to move in aqueous environments, and the bacteria which does not have this flagella may not be able to move. So bacteria, flagellated bacteria, they are motile, and bacteria which do not have this flagella, they are non-motile. So bacteria may have this flagella or may not have. The bacteria which contains this flagella. Now in this diagram I have shown only one flagellum, but one bacteria, it may have more than one flagella. So this bacterium, it may have multiple flagellas which are bound here, 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 everywhere. That means the cell wall is, or the whole body of this bacterium is surrounded by flagellum. Sometimes there may be only single flagellum, sometimes there may be two flagella or more than two flagella at one end. So likewise, there are different arrangements which are called as flagellar arrangements that we'll study later on in detail. The so this is the outermost component in flagellated bacteria which is present outside the cell wall. This is a cell wall which is shown in green, the outermost layer of most of the bacteria. Again, there is a category of bacteria which do not possess this cell wall. Such type of bacteria are classified under Mycoplasma. So there is a class of bacteria which do not contain this cell wall. They contain only cell membrane as the outermost layer. Such cell wall lacking bacteria are called as Mycoplasma. Now again it should be remembered that not all bacteria contains cell wall. There is a class of Mycoplasma which do not contain cell wall. The bacteria which contains cell wall, it may be of different composition, depending on which there are two classes of bacteria, gram positive bacteria and gram negative bacteria. So this difference is on the basis of differences in the composition of bacterial cell wall. Then some bacteria have this types of small appendages. You can compare the length and size of this with size of flagellum. Flagellum is approximately 50 to 100 times longer than the length of bacterium. 50 to 100 times longer. And in comparison to the size of this flagellum, this is the size of this appendage. This is called as pilus. Sometimes it is called as fimbriae. We'll learn the difference between pilus and fimbriae later on. So a singular appendage which is present external to the cell wall, it is called as pilus. This pilus may be present in multiples. Now they are called as pili, Pili. So a single bacterium, it may have 10 to 100 pili or fimri, which has specific function that we'll learn later on. So these are pili or fimri. Then this is, which is shown in orange color, this is cell membrane which is present internal. So it should be remembered that all bacteria may or may not present these pili. Cell membrane is present in all the types of prokaryotes, whether it is cell wall containing bacteria or cell wall lacking bacteria. Plasma membrane has to be there, which is principally made up with phospholipids. The structure of phospholipids we have already studied. This is something, one more layer which is present outside the cell wall. So this forms a covering over the cell wall of certain bacteria. It should be again noted that this external covering on the cell wall of a bacteria may be present or may not be present. So there are certain bacteria which contains this external covering. This is called as capsule or glycocalyx. So this may be, this covering, it may be structurally organized and bound to cell wall, which is called as glycocalyx, or it may be loosely bound to the cell wall. In that case, it is called as slime layer. So capsule is of two types: glycocalyx, which is firmly bound to cell wall, which is structurally organized, and it may be a loose binding to cell wall, that is called as slime. So this may be present or may not be present in certain bacteria. Now the internal components. This is the cytoplasm. Cytoplasm is a solution. That means it is a mixture of different chemicals. Along with that, this cytoplasm, it contains several important cellular components which are very necessary in order to carry out metabolic processes and growth associated functions of bacteria. So this is cytoplasm, which contains water and different types of chemicals and certain cellular components. The major inclusion present in bacterial cell cytoplasm is this covalently closed circular DNA. So this covalently closed circular double-stranded DNA, it is called as nucleoid. So nucleoid, we can call it as a genome of bacterial cell. The number of this covalently closed circular double-stranded DNA is 1. Therefore, we call this as a single chromosome. So the chromosome number in bacterium is 1. That means it contains only one set of nucleoid or genome. So chromosome number is 1 in case of bacteria. Along with that, double-stranded DNA. So this is present in all bacteria. But along with that, certain bacteria, they may have this one more covalently closed circular double-stranded DNA. You can clearly see the difference between this DNA and this DNA. The difference is it is of low molecular weight as compared to this nucleoid DNA. So this is the DNA which is present extra apart from chromosomal DNA. So this extra chromosomal DNA, it is called as plasmid. Again, plasmid may be present or may not be present in certain bacteria. If it is present, then it gives or it imparts an additional function to the bacterial cell. Cytoplasm has these components which are shown in red. These are called as ribosomes. So these ribosomes are the sites for protein synthesis, as we have learned in earlier chapter, and these are present in thousands in cytoplasm, which are essential for the synthesis of different types of peptides and proteins. So these ribosomes are of 70S type in prokaryotic cell. Then there are several inclusions that are present in bacterial cell. Ribosomes are present in all bacteria. Now, this one, these are the inclusions which may be present or may not be present in bacteria. These are metachromatic granules or volutine granules. These may be present in some bacteria and may not be present in some bacteria. So they are not essential for the growth. These are the accessory inclusions which are present in bacteria, which is required for some extra functions that we'll learn later on. So these are metachromatic granules. Likewise, there are other inclusions which are called as lipid granules or PHB granules, polybeta hydroxybutyrate granules. So these are again the inclusions may be present or may not be present in certain bacteria and which are responsible for certain extra functions of bacteria. So these are lipid granules or PHP granules which acts as a storage of lipids. So it has a function of storage. Then certain granules, accessory granules, which may be again present in bacteria, which are called as glycogen bodies or glycogen granules. So these are responsible for the storage of glycogen. Glycogen is made up of glucosinase, so it is a storage of carbohydrate. Again, this may be present or may not be present in certain bacteria. Then this is a very typical structure inclusion which is present in bacteria. Now you see all these inclusions are not compulsorily present in all bacteria. It is dependent on the habitat in which these bacteria are living or they are existing. Bacteria which are present in environments rich in metals, they have these types of inclusions. We can see structurally organized metal ions. These are called as metallosomes, and these are made up with metal ions, very small metal particles which are called as nanoparticles. So these nanoparticles are accumulated inside the bacterial cell cytoplasm, and these are called as metallosomes, which is the accumulation of metal ions, accumulation of nanoparticles. So these are not present in all bacteria. These are only present in certain bacteria which are inhabiting the environments which are rich in metals. These inclusions are called as carboxysomes. These are only present in certain photosynthetic bacteria. Carboxysomes are responsible for carbon cycle. So it contains certain types of enzymes which are responsible for the carbon cycle. Therefore, they are called as carboxysomes. These are not present in all bacteria. These are present only in certain types of photosynthetic bacteria. We can see these are again the accessory components which are present in cytoplasm of few bacteria, mostly in photosynthetic bacteria. We can see these are the cylindrical shape sacs, and these sacs are filled with gas. These are called as gas vesicles, and these gas vesicles contain gas which helps in the floating characteristic of bacteria. So particularly they are present in photosynthetic bacteria which are present in aquatic environment or in water. So it helps these bacteria to float up to a particular depth in water, and this function helps in the absorption of light rays. So bacteria can float up to a depth or up to the top of water wherever the light rays are penetrating, which are responsible for photosynthesis function. So that is about the gas vesicles present in photosynthetic bacteria. Again, they are not present in all bacteria, they are only present in photosynthetic bacteria. So these are the different cellular components. Now, there is one more very typical cellular component which is only present in endospore forming bacteria. So this inclusion is only present in endospore forming bacteria, and this inclusion is normally not present, but it is formed during the growth of bacterium, and it is formed under certain conditions. For example, this is endospore. This endospore is normally not present in bacteria, certain bacteria, but if this bacterium is present in or it is exposed to certain unfavorable environmental conditions. Unfavorable means the conditions which are not suitable for growth. So that time, in order to survive in that particular environment, these bacteria produce or they get stimulated to produce this inclusion. This is called as spore. As it is present inside the cell, it is called as endospore. Endo means inside. So this endospore is formed inside the cytoplasm after this bacterial cell is exposed to unfavorable environmental conditions like increase in temperature, absence of water, exposure to certain toxic chemicals, etc. etc. So this endospore is formed, and this endospore then takes some of this DNA inside it, and then it grows, and as a result of its growth, this endospore, so due to exposure to external unfavorable conditions like toxic chemicals, absence of water, this whole cell is destroyed, and this endospore is released out. So one cell, it gives rise to only one endospore, and this endospore, it has ability to survive under such unfavorable environmental conditions. So it survives, and whenever the favorable conditions are back, again this endospore, it gets converted into this vegetative cell, that is called as germination. So we'll be talking about in detail regarding all these cellular components in next few lectures.